Welcome to TSAT. Today I am going to discuss about virus and vaccines. So, in the case of virus and vaccines, where we have passed through this pandemic era, where we have suffered with COVID-19. So, in this context, we need to know what a virus is and how vaccines are being made. So, in this context, we need to know the nature of the diseases which are being described in terms of pandemic, epidemic and endemic. So, how they are different. In the case of pandemic is an epidemic occurring on a scale that crosses international boundaries. So, one of the basic characteristic feature to call it as a pandemic means the disease must happen globally. Cutting across international boundaries is being described as pandemic. Epidemic, affecting many people at the same time and spreading from person to person in a locality where the disease is not permanently prevalent. So, it is the fundamental difference between pandemic and epidemic is pandemic is present throughout the globe at the same time, whereas in the case of epidemic, it is a local, in a defined geographical region which is not prevalent throughout the globe. That is what an epidemic is. In the case of endemic, a disease that is constantly present to a greater or lesser degree in people of certain class or in people living in a particular locality. So, it is present in a particular locality permanently, whereas epidemic is not a permanent feature. So, endemic is locally permanent, endemic is locally but not permanent. So, this kind of difference we must be able to make to understand certain questions in this pandemic era in terms of the difference between pandemic, epidemic and endemic. So, virus was first identified by 𝗬𝗏 and by Bergenix. Initially, these are the Russian scientists who have identified what a virus is. So, the first virus which has been identified is the tobacco mosaic virus. So, based upon the shape, viruses are being classified. So, helical in terms of their shape, polyhedral, spherical, helical, complex. So, these are the different varieties on the basis of the structure they have got. Viruses are being classified into. For example, the COVID-19 virus belongs to spherical variant and bacteriophage is a virus. It is known as a bacteriophage because it infects bacteria. A virus which infects bacteria and causes disease to bacteria is been described as a bacteriophage. So, based upon the shape, this is the way the viruses are being classified into. So, to understand what virus is further microscopic or much more smaller when relatively compared with bacteria. And the structure of the virus inside is not complex as the structure of the cell in the developed organisms like eukaryotes. Virus is considered to be a link between living and non-living being. Virus has not been given a status or a position in the classification of life because it behaves as a non-living being when it is out of the host, and it behaves as living being only when it is inside the host. So, that is the reason why virus has not been given the status of a living being completely. It is considered as a link between living and non-living being because it behaves as both depending upon the conditions. The structure of the virus is simple when relatively compared with the cell of the eukaryotic cell. In the case of a eukaryotic cell, there are many number of cell organelles present in the cytoplasm like nucleus, endoplasmic reticulum, ribosomes, all these cell organelles, mitochondria have specific function to survive. Such kind of cell organelles are absent in virus, especially the ribosomes which are essential to synthesize proteins are absent. So how the virus can replicate, spread itself without these cell organelles? That is the reason why a virus infects plants and animals, causing diseases using the material energy of the other organisms, and they replenish and develop and causes disease to different organisms. So let us examine how this structure of the virus exists and how this infection is being caused by virus causing different kinds of problems, and how vaccines are generated to this is a question. So in this context, virus has got a very simple structure. It has got mainly three components. One of the main component of the virus is the genetic material. The centrally located strain, a chain coiled inside, is the genetic material. The genetic material of the virus can be RNA or DNA. The viruses which have got RNA as genetic material are called as retroviruses. So retroviruses are considered to be more dangerous. Why is it so? I will be discussing with you in the upcoming discussion. So one of the main component of virus is the genetic material. The second main component of the virus is protein coat capsid. So see this, the yellow color lining being given here, demarcated to differentiate, is a tough layer protecting the genetic material inside. And the third component is the antenna. We call this as spike proteins or spikes in the case of COVID virus, which we have experienced these days. An antenna, a protective coat, a capsid which is very tough. There are certain viruses even which can resist very high temperatures around 200-300 degrees Celsius, and they can survive even because of this kind of very tough coat the viruses have, and the inner genetic material. So these are only the three components with which a virus is made up of. And in the case of a eukaryotic cell, where you have got different complex cell organelles like ribosomes, endoplasmic reticulum, Golgi apparatus. Each and every cell organelle has got a specific function. For example, the function of ribosomes is to synthesize protein, and the protein being synthesized is used to construct the daughter cells or cell organelles or cell repair. So the synthesized protein and fat from the endoplasmic reticulum and ribosomes are being stored in Golgi apparatus. So these kind of cell organelle structures are absent in the case of virus to perform these. So because the virus is lacking these cell organelles to perform certain activities or the metabolism in the cell, the viruses depend upon the host cells for these activities. By infecting the host, causing diseases, they reproduce. So how it happens, let us examine. So in this case, the virus, for example, the COVID-19 virus, when the coronavirus, when it infects, the structure with which it attaches to the cells, for example, consider this as a human cell and this as a virus. So it is attaching itself to the cell with the help of spike protein. So with the help of spike protein, it is attaching itself to a cell. After attaching, it introduces the genetic material to the cytoplasm. So after introducing the genetic material to the cytoplasm, from the cytoplasm, the viral genetic material might be RNA or DNA. It is RNA in the case of COVID-19 virus, which is described as a retrovirus, enters into the cytoplasm. From the cytoplasm, it enters into the nucleoplasm and it incorporates into the genome of human beings. So after incorporating here, because the basic thing which we need to understand how it replicates is the incorporated DNA, the incorporated genetic material, the viral genetic material into the human genome is being considered as the human genome by RNA. So what happens is, from the cytoplasm, for the purpose of protein synthesis, so this is the cytoplasmic region. We have got different cell organelles like ribosomes, endoplasmic reticulum. So the information to synthesize protein is in the genetic material in the nucleoplasm. From the nucleoplasm, the information is being carried to the ribosomes in the cytoplasm by mRNA. The mRNA, after entering, which can enter into the nucleoplasm, means inside the nucleus, carries the information in the genome and informs the ribosome to synthesize protein. But the problem after infection is, because the viral genetic material has been incorporated into the genome of a human being, the information being carried by mRNA will be viral information. This viral information given to ribosomes synthesizes viruses rather than human protein. In this process, the cell materials, proteins, fats of human body are being used to make viruses. So what happens is, by using all these materials of our body, when viruses are being made in the cytoplasm, after a certain amount of viruses load being created, the cell bursts and releases viruses into the bloodstream. This outburst of innumerous viruses into the plasma of the blood leads to a condition known as a cytokine storm. So cytokine storm is a condition where which is because of a secondary infection. So the first infection is known as the primary infection. After it is multiplying and bursts out into the body, into the plasma, and millions of viruses enter into the blood plasma, and after the viruses are considered as antigens or foreign bodies by our body. So to counter this, antibodies are being generated by the lymphocytes, or some molecules are being produced, protein molecules, and some cells like T lymphocytes produce antibodies. So there is huge volume of certain molecules and cells being produced in the plasma, more than the essential, where the plasma will be overloaded with these viruses and antibodies. So the capillaries which are so thin will be blocked by these more cell organelle structures or cells or antibodies and antigens. When the capillaries are being blocked by this material, it leads to the arresting the supply of oxygen to different organs and organ systems, leading to a condition known as multi-organ failure, and will lead to the death of the patient because of cytokine storm. This is the main reason for the death of the patients. So to counter this condition, we have developed vaccines to viruses. So vaccines consists of disease-causative organisms. They boost the immunity. So what do you mean by this? So what vaccine contains is, vaccine contains microorganisms causing diseases. But the microorganisms causing diseases are injected into you not as it is. They are attenuated or killed, means they are weakened, or the dead virus is been introduced into you, so that the disease will not be manifested so seriously. Or sometimes a part of the viruses are being introduced into our body, known as subunit vaccines. So different technologies are being used in this pandemic era in terms of manufacturing vaccines, which we will be discussing. So in this process, vaccine contains disease-causative microbes which are not introduced into you, injected into you as it is. They are being killed, attenuated, and they are being introduced, so that the immune system defends it very easily and will be able to know about the pathogen and makes antibodies. Antibodies are weapons created by your body by 𝗹𝗶𝗁 to attack the viruses, to make you disease free. So here in this process, what we do is, we need to know what a vaccine is. Vaccine is a substance used to stimulate the production of antibodies and provide immunity against one or several diseases. So what do you mean by this? A substance to stimulate the production of antibodies. So what are antibodies here? Antibodies are cells or molecules produced by lymphocytes. A lymphocyte is a variant of 𝗹𝗶𝗁, produces a temporary cell against a specific pathogen like virus which has entered into you. So this vaccine, which is not a which contains a virus causing disease not in an active state, stimulates the immune system, and the immune system will be able to know what kind of antibodies need to be made in a real-time infection. Already the immune system will be given information how to make weapons against the virus. And this vaccine will not be able to create disease because the introduced viruses are in a dead form or weakened form, where your immune system will be able to tackle the artificial infection. So, when your immunity knows how to make antibodies, means weapons against this, you will be getting immunity to these diseases. So, prepared from the causative agent of a disease, its products are synthetic substances. So, need not be a complete virus, a part of a virus, or synthetic substances like RNA treated to act as an antigen without inducing the disease. So, this is the antigen is a foreign body. So, by introducing this a foreign body known as a virus or a part of a virus, initiates the immune system, that is 𝗹𝗶𝗁, to create an antibodies, means a weapon. So that in a real-time infection, the antibodies which are already been initiated or generated because of an artificial infection known as vaccine will be able to defend you from the disease very easily, because the body need not make antibodies after the infection. After real-time infection, already there are antibodies to attack the real infection, so that you will be saved from the disease. This is the basic principle involved in the case of vaccine. A vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. So, you will be acquiring immunity to a disease. A vaccine typically contains an agent that resembles a disease causing microorganism. So, a vaccine contains a disease causative microorganism, and it is often made from weakened or killed form of the microbe. So, that is how it is been introduced, or some surface protein like spike protein is just introduced to initiate the generation of antibodies. So, for example, see, this is an antibody which have developed a cup-like structures to trap the spike protein of COVID virus. So, this is a kind of temporary cell structure being created by a lymphocyte known as antibody to attack the viruses, hold the spike protein of the viruses, make them dysfunctional. So that because spike protein is the one which is attaching to your cells to introduce the genetic material, and the spike protein itself is being made dysfunctional, they cannot transmit the genetic material to proliferate in your body. That is a basic mechanism being involved in the case of vaccines. So, this constitutes the immune system. So, immune system consists of the 𝗹𝗶𝗁, which is present in your blood, which are been classified into granulocytes and agranulocytes on the basis of the granules present in the cytoplasm of 𝗹𝗶𝗁. In these agranulocytes, there is a variant of 𝗹𝗶𝗁 known as lymphocytes. The lymphocytes, out of these different lymphocytes, T lymphocyte is a variant which is responsible for generation of antibodies. So, this constitutes the immune system of our body. Apart from these cells, the other line of immunity to our body is the probiotic bacteria in symbiotic association with you on your skin and in the intestines, protect you from harmful microorganisms, is also a line of defense or immunity to you apart from 𝗹𝗶𝗁. So, in these different types of vaccines which you have come to know, there are certain different kinds of vaccines work under different technology. So, we need to know what are the different kinds of vaccines used in treating COVID-19, what is the technology involved, who is the producer, we need to know. So, Covaxin is being produced by Bharat Biotech International Limited, which is associated with the Indian Council of Medical Research. So, this is we are proud to say that this is an institution or an organization existing in our city, Hyderabad. So, there might be some information you need to present about Covaxin or data. Covishield is being generated by Oxford AstraZeneca. The technology involved in the production of Covishield is different from Covaxin. How is it? We will be discussing with you. and even in the case of Pfizer, developed these Moderna, etc., depends upon technology known as mRNA technology. So, these are some of the examples of vaccines of COVID-19 developed with the different technologies. What are they? We will be analyzing. So, based upon the technology, where vaccines are being classified. So, in what way these vaccines are being classified? The live attenuated vaccines means the virus which is being introduced into you will have life, but they are weakened. They will not have the capacity to generate a disease to you. The next type of vaccine is inactivated vaccine, means completely killing them. The virus is being killed and it is being introduced to boost to activate the immune system. Subunit vaccines, the whole virus is not been introduced into your body, only the spike protein is a subunit. For example, in the case of Covishield, a part of the virus has been introduced to activate the immune system, are known as subunit vaccines. Next is viral vector vaccines. Is a viral vector vaccine. In the case of viral vector vaccines, virus is being used as a vector. A vector is one which carries something. So, here for example, in the case of Covishield, what is being done is an example of viral vector vaccine. So, in the case of viral vector vaccine, the vector is a virus known as adenovirus present in the chimpanzee. So, we have selected the vector as an adenovirus present in the chimpanzee, which carries the subunit of COVID-19 virus. So, the spike protein of COVID-19 virus is attached to this virus known as adenovirus acquired from a chimpanzee. So, why we have collected adenovirus? Because this adenovirus does not cause harm to you. And why? What is the necessity of virus? Because virus nature is infecting you and proliferating in you. But this is a virus which does not cause disease. In this process of spreading of the virus, it carries the spike protein attached to the virus. So, in this process, it activates the immune system to generate antibodies to give certain protection, which is known as viral vector vaccine. So, mRNA vaccine, with the Pfizer and Moderna are the examples of mRNA vaccines. So, in this case of mRNA vaccines, as I have told you, mRNA is the nucleic acid sequence which contains these nucleic acids like adenine, etc., guanine kind of things, are the nucleic acids which are present in RNA. With this code, it enters into the cell nucleus and gets the information from the DNA and transmits it to the ribosomes in the cytoplasm. So, and this information code, mRNA is carrying a code to carry antibody in a lymphocyte. So, for example, a lymphocyte is a cell. Lymphocyte has got nucleus. The nuclear DNA of the lymphocyte has got an information to make an antibody, which is been carried by mRNA from the T lymphocyte nucleoplasm to cytoplasm of lymphocyte. So, if you are able to decode this and synthesize this mRNA in the laboratory, which can be injected to stimulate the immune system to generate the essential antibodies, because the mRNA code requires the information to manufacture antibodies. So, you are directly copying the code to synthesize protein of antibodies, consists of mRNA vaccines. So, these are the most common live attenuated vaccines. These are the different examples of vaccines which are live attenuated. So, you might be able to give some information regarding these. What are the different examples of live attenuated vaccines? The smallpox vaccine, or the chickenpox vaccine, or the yellow fever, or Japanese encephalitis, or polio, or measles can be given as an example of a live attenuated vaccine. So, Covaxin is a kind of vaccine where inactivated vaccine, in this case the whole which is been developed in our city Hyderabad, by Bharat Biotech Limited, where the whole virus which has been inactivated is been introduced, is one of the classic example here, which is one of the best vaccines to develop immunity to you. So these are some examples of conventional vaccines. So in the case of Covishield, we have taken a vector as chimpanzee adenovirus, and you have attached a spike protein to this adenovirus, and this genetically modified spike protein attached virus is being synthesized in the laboratory and used as a vaccine. So in this context, we need to know what is meant by a retrovirus. So classic example is the coronavirus is an example of retrovirus, and even HIV is an example of retrovirus. So what is retrovirus? Viruses that carry reverse transcription are called retroviruses. These viruses can convert their RNA into DNA copy. This process is catalyzed by reverse transcription enzyme. So what is happening here is, when the virus has got RNA as genetic material, this RNA cannot be incorporated into our genome, which is in DNA. So retrovirus has the capability to transform the RNA into DNA and incorporate into our genome, misguiding the mRNA of our cell as the information of human being, but in this process it transfers the information of viral RNA information to synthesize viral proteins. So this is how retroviruses are considered to be more dangerous, because in the process of reverse transcription, what happens is there is possibility of more mutations. Mutations are responsible for a variant of concern. A variant of concern is a mutated virus which can impact a wave of disease. A first wave or the second wave or the third wave is because of a variation of concern. Means in this process of reverse transcription, what happens is the code of nucleic acid sequence will not be exactly replicated. Sometimes an additional nucleotide might be added or deleted. In this process, the gene code varies. The varied gene code may lead to turn into a lethal mutation, means a dangerous mutant variant of concern, which may lead to create another wave of a disease. So that is the reason why retroviruses are considered to be dangerous, because they use RNA. In this process of reverse transcription, there is possibility of more mutations. Mutations may lead to a variant of concern, means which can cause another wave of disease. So cytokine storm we have discussed. Cytokine storm is the abnormal release of viruses and antibodies and protein molecules to counter the virus attack, and clumping up of these leads to blocking of capillaries, leading to arresting of oxygen to the organ systems, leading to failure of organs and death. This is what about virus is, and we are going to meet in the other session discussing a different topic in science and technology. Thank you.